So hi everyone, welcome and thanks for joining us. Today we're going to be talking about progress that we've made in the area of cellular pathology and its application to clinical practice, hopefully in the foreseeable future. So what is cellular pathology? Cellular pathology is one of the fundamental principles of our field. It was coined by this gentleman here, Rudolf Virchow, where he postulated that all diseases are not merely characterized by their clinical symptoms, but by typical cellular and anatomical changes that we see under the microscope. And although we take this for granted today, this was a paradigm shifting idea 150 years ago when he came up with with this hypothesis. So how does this look like in regular day-to-day practice of a pathologist? This is a section of cortical brain tissue. And as you can see, there are three major cell types that make up the human cortex. You have these larger cells, which are your neurons, that have a large nucleus with a prominent nucleolus. You have your medium-sized astrocytes that are somewhat irregularly shaped. And then you have your oligodendrocytes that have a perinuclear halo or clearing around their nucleus. And it's these cell types that provide the cellular components of our amazing central nervous system. Now, of course, as the hypothesis would state, when there is a disruption in this architecture, there is disease. And this is an example of that. This is a ganglioglioma. This is a tumor where you have proliferation of the neuronal components. And you can see that we have this abundance of these larger cells with a prominent nucleolus reminiscent of the neuronal components of the normal brain. Thankfully, these are curative with surgery and thus are assigned a WHO or World Health Organization grade 1. Here is another disruption in the anatomy. This is an oligodendroglioma, and you can see why this was named as such. It has that perinuclear halo around all the tumor cells that are reminiscent of the resident oligodendrocyte. These tumors are in fact fatal, but have a relatively indolent course compared to other brain tumors where patients live 10 to 15 years and are graded as a WHO grade 2 or 3. Lastly, and probably the most common tumor that we see in our practice are astrocytomas. These have, again, the proliferation of these atypical cells that resemble resident astrocytes, and these tumors tend to have a much more aggressive course. So you can see that by understanding the normal histology and how it can change, we are able to diagnose not only abnormalities and disease as Dr. Virchow postulated, but we can also prognosticate patients based on the different patterns that we see. Now, how do we memorize these patterns and do this in a reliable and objective way? Everyone has their own secrets. This is mine. I tend to try to relate them to objects I see on a day-to-day basis. So when I look at neurons, I think of the large eyes of an owl to represent the nucleus and nucleolus. When I see fried egg appearing cells, I think of a oligodendroglioma. And when I see these atypical cells irregularly arranged, I think of a pile of potatoes. And this, of course, helps us add some objectivity to this relatively qualitative task required to subclassify these tumors. Now, this is an example of interpatient heterogeneity and patterns that, again, have prognostic significance. There's, of course, also intrapatient heterogeneity that we may see within a tumor. This is an example of palisading necrosis where we see the cells accumulate around an area of necrosis, and what we're in fact seeing right here is a hypoxic area of the tumor. When we see these in tumors, these have suggest a relatively aggressive phenotype of necrosis, and thus patients with these are enough to grade the patient a WHO grade 4, which has a much more guarded prognosis for the patient. So very, very versatile tool, both for inter-tumor variability and also intra-tumor heterogeneity, and thus it's still a central tool in our modern medical practice. This is kind of a comparison to other tools that we use for diagnoses, and of course, these are found in a variety of different flavors. You have an inspection that the primary physician can do on the patient to find lesions or stigmata of specific genetic disorders or tumors. What we see here are neurofibromas on the skin, which is a pathognomonic feature of neurofibromatosis. There's, of course, imaging modalities that can also be done in the emergency room at the point of care at the bedside, which are useful, but of course are relatively qualitative and provide more of a differential diagnosis than an actual actionable diagnosis. On the other side of the spectrum, we have global molecular technologies such as sequencing that can provide very, very precise mutations. And these are, of course, of high diagnostic value as we move into the era of personalized medicine. But as you can see, these come at a high cost and at the moment, a relatively prolonged turnaround time. Right in the middle is where I think cellular pathology continues to excel in our medical practice. It is a tool that is both relatively cheap. It's a tool that can be prepared on the order of minutes intraoperatively or on the order of days for most routine cases. And as you can see, provides more objectivity than some of the bedside tests. So I think it's a very important tool and will continue to be so moving forward. So we need to improve it, though. We can't. We don't want to be in a position where we use tools that are unchanged for hundreds of years. So how can we improve this precious tool that we have in our toolbox? And the emerging approach is to use computers and the precise nature of mathematical models to better model how these patterns change both between and within patients. And the tool of choice that has emerged are what's called neural networks, which are computer algorithms designed for what's called computer vision that have been inspired by no other than the human brain. So this is kind of a pathway of how we see. We see the orange circle, which is our eyeball, and when light hits a cat and is emitted to our eye, the shape of the cat will activate our photoreceptors in our eyes, which will send signals into our primary visual cortex. Here, the chief signal is broken down into edges and lines and color, which are then integrated into higher order, secondary order neurons, which will take those edges and start making more complex objects like faces, whiskers, cats, nose, tail, and then the objects will become more complex as we go into higher order centers of our brain. And then eventually our sensory system will appreciate that we're looking at a cat based on the pattern of features that the retinal cells have detected. So the basic idea of these computational neural networks is to replicate this amazing process by having multiple layers of neural-like elements that can detect initially very elementary shapes, edges, color, and then as information is passed on into deeper layers within the neural network, more complex and diagnostic patterns can be generated, and then eventually the computer will use the high-level features that are constructed late in the neural network to make a specific classification. So we're going to use, I'm going to use some examples from our group to highlight how neural networks is really redefining how we practice cellular pathology. And really the talk is broken up down into two different sections. We have a supervised learning section that we will start with that's a little bit easier to wrap your head around. And then we'll go into some more exciting stuff on unsupervised learning and how we think that it's going to play an important role in this field. And we'll again talk about the things that we saw with the regular pattern recognition tasks, such as can we resolve intra and interpatient heterogeneity. So this is what the workflow looks like for a typical supervised deep learning workflow. It's pretty much how you could imagine we learn. You collect a large number of images. These images that we use to train them are approximately 516 microns in length, so 516 microns squared images, and that size is a very nice size because it captures a lot of the repetitive patterns that we see under the microscope. We generate hundreds of thousands of these images. We label them based on the diagnosis that we want the computer to learn, and then we run them through these neural networks in a process called training. What the neural network does during this process is it takes each image and looks for the lines and edges to break the image down into digital data, and as it passes that information through the neural network, it will start making more complex objects such as circles and ellipses, and it'll use that information to make more complex objects as it moves the image through these different layers of the neural network. At the end, it will try to assess if certain patterns that it generated have any diagnostic value to separate that specific class with other ones in the training set. And doing this iteratively for each and every image, it will generate an optimized neural network where the specific connections have been weighted to and optimized to allow making these classifications in future images. So when you present to it a new image or a test image, it will run this image through the neural network. It will see which specific neurons it activated, and then it will use that information in the final penultimate layer to generate high-level representations that have diagnostic value. And then in the final layer, it will output one of the classes in a probability score-based readout that will help guide you into what most likely this case is. And this, of course, has recently been shown to meet expert level-like performance in a number of domains, including both pathology and medical image analysis in general. So this is what it looks like in our hands. These are full whole slide images that we that pass through our medical practice, and we can take these images and train the computer to recognize different components of the slide, so different tissue types. And you can see that once it's trained and these slides are run through, you get these beautiful pseudo-color images that accurately annotate the different components of the slide. And by doing this, we now can not only annotate the intra-tumoral heterogeneity, but we can quantify it in a very, very precise manner. And so this is really, I think, a beautiful example of what supervised a supervised workflow looks like for assessing intra-tumor heterogeneity. Just to show you how precise this can be, this is a section of a glial tumor, and you can see that the tissue has been fragmented into many tissue components. We run this slide through the neural network trained to recognize specific properties that are of diagnostic value for glial tumors, and you can see that it only found the glioma in certain components of this slide. So this is really nice because now we can compare it to a ground truth or the kind of precise location of the glioma using immunohistochemical or molecular techniques, and you can see that the fragments that immunohistochemically stain for this mutation found in the glioma resemble the pseudo-color map that this neural network generated to annotate the image. Of course, you can zoom in, and what you see when you zoom in, you can see again the ability to capture some of the intratumoral properties of this tumor. So as we see here, brown representing tumor and pink representing normal brain, we see that this is an infiltrative edge of the tumor, which is nicely demonstrated by this neural network. Of course, you don't only need to do this for H&E stains, you can also do this for immunohistochemical staining. This is a MIB1 proliferation marker, which is used in many tumor types as a marker of the proliferative capacity of the brain of the tumor, and of course has prognostic value in many tumor types. So what we usually do in many cases in our practice is we try to estimate it based on our kind of experience with this marker. So in this case, I would probably estimate it somewhere between 35 and 45%. As there are areas that are very, very high in proliferation and areas that are also understandably much lower. And you can see the problem in these types of global estimations is that we don't capture the full heterogeneity or biology of the tumor because, again, not all regions of this slide are proliferating equally. So when we run it through a neural network trained to recognize different proliferation indices, you can see that not only do we get a nice pseudo-color map of where the proliferative capacity is on this slide, but we also get a distribution score for the global average of the slide, but also it can break it down into different percentages in different regions. And I think that is a really, really important way to start documenting this data in a much more advanced and complex way. So that's really kind of an introduction. Hopefully that's our supervised learning for intra-patient heterogeneity. We can now move on to look at how these tools are being used to resolve inter-patient heterogeneity, which has, of course, the most concrete application in our current clinical workflows. So one of the major tasks that I'm asked to do as a clinician is to take tumors resected by our neurosurgery colleagues and classify them into the most appropriate tumor class. And this has important implications, as we saw, as some tumors may be curative, other tumors may be much more malignant and require specific types of therapies. Of course, there are non-neoplastic entities that we also have to entertain because they may be better treated with antibiotics or other non-chemotherapeutic strategies. So we use the same approach. It's very straightforward, but instead of specific tissue types, we're going to introduce different tumor types to the training data and the labels that we provide for the computer, and it will painstakingly again go through these hundreds of thousands of images and try to correlate what diagnostic entities the human expert has labeled with specific patterns that are available under the microscope. We train it, and then once we have a trained CNN or a convolutional neural network, as these specific neural networks are called, we can present to it new images and try to classify them in a way that has some actionable and diagnostic value. So we're constantly doing this, as this is kind of the major goal of computer vision in computational pathology at the moment. And this is one of our first large-scale experiences with this approach. We collected 2,257 slides spanning 400 cases. Just to give you a context, that's about 0.75 of a full-time equivalent of one of our neuropathologists. So quite a significant number of cases. And for each case, we're going to break down these slides into these diagnostic 516 micrometer square sections, and then we're going to run those through the neural network, classify them, and then try to localize where the tumor is on this slide. And then once we've identified the lesion, we have a second neural network that takes those lesional slides shown here in black and then tries to classify them and summarize them in a clinically useful way. So we do this with one click of a button for 2,257 slides, and this is what the results look like. This is a plot that allows you to compare the distribution of human diagnoses to computer predictions. And you can see a few things. There are different frequencies of different tumors. The major tumor types are schwannoma, glioma, meningioma, and metastatic carcinoma. And you can see for the most part, for these common tumor types, we do extremely well. One of the nice things that we've done to help reduce errors is create this non-classified class that allows the computer to triage challenging cases for human review without having it to have to guess. And that's really reduced the number of errors that we make. It's suffice to say that this kind of version 1.0 of this exercise, we consider it quite successful. We get about 90% of cases correct based on comparison of the computer and the human diagnoses. And we're still trying to work through those discrepancies to further improve this in new versions. Reasons for errors include diagnostic mimics that are very difficult to resolve just on H&E stains. So one of the things that we're doing is trying to incorporate molecular data into these classifiers to further reduce the errors. But given that there was no molecular information, there was no clinical information, I think this is a strong starting point towards implementing these tools in our clinical practice. So one of the nice things about working in this field is that we need to try to reimagine how we document and present data to clinicians. So this is kind of our standard neuropathology report for those of you who see these on a day-to-day basis. And usually they're generated in a word processing file, so they are quite wordy, as we have to describe exactly what we see, whether it be fried eggs or potatoes. We have to write that in some sort of medically appropriate manner. But we think that by doing this digitization of the data and using these computational tools, we now have a much more visually appealing way to document our medical findings in a microscope and do this in an automated way. So we're trying to generate these reports that are automatically generated by computers, where physicians and pathologists can approve and confirm the findings, and it just documents it in a much nicer way. So this is one of our recent kind of templates, just to show you we're always improving it based on feedback we get from pathologists and clinicians about what they would like to see on these pathology reports. On the first page, we try to mimic what's already working. So we have a kind of a demographic page that includes patient information, clinical information that's provided by the clinician. In our summary, this really replaces our diagnostic line where we have a diagnosis. So this is a glioma. Here we also provide counts. So if a case had multiple slides, it will tell you how many times and how many of these slides were classified as gliomas. And then here we have a recommendation about what the kind of cutting edge standard of care would be for a glioma at our institution. So if someone at a remote center was using this tool, they will be provided with some recommendations about what the next steps would be to appropriately work up this case. Page 2 is meant to document a lot of the stuff that we now type out. So you have the original slide. It's very evident what was provided to the physician. And then you have these nice pseudo-color maps, as we saw, that kind of document where the tumor was and where the computer sampled in these green boxes to make the diagnosis. We have slide breakdowns to kind of document exactly how much and what types of tissue were detected. We have probability-based descriptions, predictions based on probability scores, a traditional approach, and using dimensionality reduction approaches that we've developed internally. You have an approximate surface area of the lesion that can help guide molecular testing, and then we have a kind of high-power view of the lesion to again document this in a very visually appealing way. We have quality control metrics just to kind of ensure people know what tool was used to make these diagnoses, but we think that this is the future of AI and pathology working together in the future. This is one of our immunohistochemical reports, and as you can see, again, provides a traditional overall score on the first page, but on the second page, again, you get these documentations of where the computer detected high proliferative activity, and then you have this pie chart breaking down the percentages of proliferation indices rather than just giving you an overall score. And these may in fact turn out to be a more accurate representation and prediction of how the patient tumors behave. These tools are very, very nice, but the beauty of these tools, of course, is that you can now put these in the cloud. You can make them cloud-based tools, and that's exactly what we're doing here. This is one of our sites that we've just launched that allows researchers that are interested in testing out this technology to log in. We have different apps for different projects, and you can use these apps. And just to show you how this works at the very moment, here's a demo of what the apps and their functionality as of today. So a person would log into the system. You see the apps available. You click on one of the apps. If you have an image, you can drag and drop it as you would with a normal website. You can put in your metadata, your clinical metadata, the age, clinical history of the patient. You can choose which model you want to use. We currently have 2 models, one for brain and one for ovarian cancer. You submit the case, and the case is sent to our cloud. It's analyzed by these neural networks, and then you get an email that when the report is complete, and you can download these reports that I've been showing you for your research and projects. You can, of course, eventually when these are validated, they're meant to also be used clinically, but of course, this is a research-based tool for the time being. But just to give you a sense of how automated this process can be, which I think is very, very empowering, given the cost of this test, given the ubiquitous nature of pathology slides in the world. This can be done anywhere in the world. So I think that's really the power of this technology, and we are in fact using this tool to now do external validation at other centers. So hopefully that gives you an outline of our supervised learning initiatives and their relevance to our clinical practice. I now want to shift gears to move to unsupervised learning, which is an area that we're very excited about in the lab, and so is the kind of machine learning community in general. This is a quote by Jan LeCun that I really like. Just shows how excited the field is about unsupervised learning, where he tries to relate learning to a cake. So he says most of human and animal learning is actually unsupervised learning, and if intelligence was a cake, unsupervised learning would be the cake, supervised learning would be the icing on the cake, and reinforcement learning would be the cherry on the cake. So you can see the relative proportions. It's very clear that what we've seen is just a tip of the iceberg for artificial intelligence and our quest for autonomous medical image analysis. So we also want to develop these unsupervised tools to really allow us to advance this field further. So this is perhaps the example that I like to conceptualize best about what the power of unsupervised learning approaches is. So imagine there is a child that's playing with Lego and you want them to learn a specific task. The supervised approach would be to generate these paper bags, put colors on them corresponding to their Lego pieces, and monitor how good they are at putting the correct color in the correct paper bag. There's some advantages to this, as we saw, is that you can quickly monitor the performance of the child on a day-to-day basis based on the number of correctly placed Lego pieces in each of these bags. But really the goal here is for them to replicate a defined task that you deem is important. And in machine learning, this is called classification, as we saw. Now, there's a lot of problems with this, of course. If the child goes to school and exchanges some of their yellow Lego pieces for some orange Lego pieces, it becomes unclear what bag that orange Lego piece should go if the defined task does not have a paper bag dedicated to orange Lego pieces. The alternative approach is not to provide the child with any direction and allow them to play with the pieces until they learn that there are some common patterns in their Lego piece collection. And eventually, I think most kids will recognize that color is a major driver of the heterogeneity in their Lego pieces and will neatly group these pieces into specific clusters or subgroups as shown here by these bins. The beauty, of course, is that if they were to exchange Lego pieces at school, they could, of course, always make an additional cluster or additional subgroup to accommodate those new colors. But I think the most exciting part is that one child may decide to group the pieces based on color, while other child may decide to group them based on shape. And so it leaves open an opportunity for discovery and anomaly detection. So if there was an irregularity in the data that was unanticipated, these types of approaches are much better at detecting these sorts of anomalies and highlighting them to both the parent or the guardian or also the clinician. So how does this work in our specific model? So this is our supervised model where we have a brain tumor slide that's run through a brain tumor educated neural network. And the idea is that it's going to try to classify it based on the different classes that we train the computer to learn. The unsupervised approach is to take a somewhat educated neural network in one domain, such as brain tumors, and remove it from its native environment and put it in a novel environment. So perhaps present to it non-CNS tumors or non-central nervous system tumors and see if it can find some sort of intelligible pattern in this novel environment. And again, because these layers have been trained, the idea is that even this non-brain tumor will be passed through the neural network, and it will, again, try to break these patterns down into lines and edges. And as it progresses the image through the neural network, those lines and edges will become more complex objects until that final layer where you have these high-level objects that will be used to classify the images. So the unsupervised approach is because these classes don't make any sense outside of the nervous system. We want to hopefully assume that that final layer where you have the most complex features are features that are shared between different tumor types. So mitotic activity, our proliferative activity is something that would be ominous both in a brain tumor and a renal cell tumor, as shown here. So we want to take advantage of these similarities and see if we can extract these final feature representations within the neural network, and instead of classifying, use clustering and dimensionality reduction approaches to see if we can find meaningful clusters in the data. So let's see how this works. So to do this, we went to the TCGA database. So we have not only unique tumors, but tumors scanned at different institutions. And we tiled these large gigabyte-sized images, again, into 516 micrometer square images. These images are run through that neural network. We collect these feature representations in that penultimate layer, quantify them, and instead of trying to classify them, we can instead cluster them using hierarchical clustering or T-SNE approaches. And you can see that there is at least some clustering of the different tiles of this image into specific subgroups and areas on these dimensionality reduction plots. So once we generate these subgroups, we can now plot them onto the slide. The null hypothesis is that this is all gibberish and that these subgroups would be randomly distributed throughout the image. But as you can see, there is in fact specific areas of the slide that are enriched for one particular subgroup than the other. So the alternative hypothesis is that it is in fact detecting a meaningful intra-tissue heterogeneity in a relatively unsupervised way. So we can actually go in and ask pathologists what do they think these regions represent, and you can see that these different clusters represent these teal areas represent normal renal parenchyma. There's this green area here within the renal parenchyma beside the lesion that was color-coded uniquely. And when we look at these areas, it looks like renal parenchyma, but in addition to that, it has what's called interstitial fibrosis and tubular atrophy, probably due to the proximity to the tumor. As we have here, we have a yellow area that represents the actual clear cell renal cell carcinoma. We have a neighboring red area that represents kind of stromal and necrotic tissue. We have orange, which is the adipose tissue beside the specimen. And then we have even a purple strip here that represents kind of inflammatory infiltrates that have come in response to the presence of this tumor, most likely. So you can see we're able to, in a relatively automated way, without additional training, take a brain tumor educated neural network that took many, many months to generate, and without any additional work, we're able to very precisely annotate this image. It's actually really interesting to go through the different subgroups to see how the computer organizes them, to see if it's doing this in a relatively intelligible way. And so you can, instead of having 8 groups as what I showed you, you can actually cycle through the different subgroups and see exactly the decisions the computer is making to form these subgroups. So here is the slide. When you go to 2 subgroups, what the computer decides to do first is separate the green, the paucicellular components of the image with the hypercellular components of the image. So you have fat, hemorrhage, and adipose tissue being grouped as green, and then you have your renal cell carcinoma being grouped with your normal renal parenchyma, which are much more cellular components. When you go to k equals 3, it obviously recognizes that there's a difference between the tumor and the normal renal parenchyma, so it divides that group first into normal renal parenchyma and RCC. At k equals 4, you can see the step that happened here is that it recognized the abundance of inflammatory cells in this area, and it separated out from the neoplastic component of the tumor. K equals 5, it took the adipose tissue and divided it into adipose tissue with and without. It took the paucicellular tissue and divided it into adipose tissue, orange, or tissue components with vasculature, so adipose tissue with vasculature, hemorrhage, and some of the edge artifact also got grouped into this subgroup. At k equals 6, it divides the RCC with some of the artifactual tiles that got subgrouped into this specific class that had some blur artifact. It separates those out. K equals 7. We see that it now took that renal parenchyma and generated the renal parenchyma with and without this tubular fibrosis, this interstitial fibrosis and tubular atrophy. And at K equals 8, it took the edge artifact, hemorrhage, necrosis, and stroma, and separated out into hemorrhage and necrosis, stroma, and vascular tissue. When we go to K equals 9, we can't see, the human interpretation didn't find any obvious separation, so that's why we chose K equal 8 to display this type of intratumor heterogeneity. Now, the beauty of this is it works in a number of different tumor types. So this is a adrenal gland, again surrounded by fat, but in the adrenal gland there is a metastatic pancreatic adenocarcinoma. We can again take this slide, decompose it into these 516 micrometer square tiles, extract the feature representations in a final layer, and then subgroup them based on similarity. And you can see this is the product. We see that fat being separated out by the red color or the red subgroup. We see the tumor in green right in the middle, but also there's some kind of magenta or purple components within the tumor. So it'd be interesting to see what those are. And then the adrenal gland here, you can see there's some layering with a deep yellow layer, an intermediate orange layer, and then a superficial blue layer. It turns out that those are the layers of the adrenal gland. So it learned to separate out different layers of the adrenal cortex. And then you can see here, when we look at the tumor, the different colors represent both the adenocarcinoma component, or it looks relatively poorly differentiated to me at this magnification. And then of course, the magenta color, we have these more squamoid-like differentiation. So again, we've resolved intra-tumor heterogeneity in a relatively unsupervised way. Here is another case from the TCGA network, from the uterine endometrial carcinoma. Again, a tumor that this computer, this brain tumor educated neural network has never seen before. You have the kind of endometrial tissue here, and then you have the tumor here that's much more purpley on this H&E slide. We again deconstruct the image into these diagnostic tiles and then try to group them. And as you can see, it did a very, very good job separating out the normal endometrial tissue into one subgroup. There seems to be some artifact or some myxoid material in this kind of the middle area of the endometrial carcinoma, and then of the endometrial tissue. And then you can see the carcinoma. There's three major colors. There is this lime color. There is this kind of red color on this fragment, and then green color here. So you can look at those under the microscope, and you can ask a kind of an expert pathologist to give you the interpretation why the computer subgrouped this relatively homogeneous looking tumor at low power into different subgroups. And in fact, the lime color is an adenocarcinoma component forming these nice glandular structures, whereas the red cluster, as you can see, is made up mostly of a poorly differentiated tumor component. And this is, we think this is quite a nice, convenient way to assess inter-tumor heterogeneity, intra-tumor heterogeneity across a variety of different tumors. And we're actually now exploring if these different tumor areas actually have different molecular profiles, which would be very, very important for personalized medicine efforts to be able to decompose tumors into different morphological subgroups and do regional molecular profiling rather than looking at the overall tumor molecular profile, which may be less informative for guiding therapy. So that's the end of our intra-tumor heterogeneity story. Let me guide you through our interpatient heterogeneity story. One of the nice things about renal cell carcinoma is that it is a very good model to study inter-tumor histomorphologic variation. There are three different types of renal cell carcinomas: chromophobe, which have a relatively good outcome. We have papillary and clear cell, which, as you can see, have a sequentially more aggressive clinical behavior. But also within the clear cell carcinoma group, as well as the papillary group, you can further grade them based on this ISUP, or International Urological Society for grading RCC tumors based on their nuclear features and their nuclear pleomorphism. So you can see the lower grade tumors have a relatively small dot-like nuclei, whereas the higher grade tumors have a much larger nucleus with prominent nucleoli at different magnifications. And then your group 4 is these very ugly sarcomatoid-looking, rhabdoid-looking cells, and you can see that grading clear cell renal cell carcinoma based on these ISUP grades have some prognostic significance as well. So 2 dimensions of interpatient heterogeneity that are of clinical value, and of course have been generated based on decades of consensus between experts about the recognition of these patterns and their clinical significance. So to see if we could do this in a relatively unsupervised way, we took 550 cases that had the same scanning parameters as our initial training model, and most of these were comprised of the TCGA cohorts, KERP or kidney tumors that are papillary and kidney tumors that are clear cell. And we asked the neural network to use these feature representations found in that final layer to group them into different classes. So you can see here the computer decided there is 2 optimal classes as shown here, green and red. And when you see the enrichment, the relative enrichment of these different TCGA classes based on these subgroups, you see that one group is primarily composed of papillary tumors, while the other one is composed of clear cell renal cell carcinoma tumors. You can also visualize this on our TSNE plot that shows the majority of one TCGA cohort being in one region of the TSNE plot, while the other ones are in the other region. So very, very good separation, relatively without any sort of training to differentiate these different subtypes. Now, how does this work? Why does this work so well? And what we think is going on is that we recently showed that as information is passed through a neural network, there is some contention if a neural network is actually learning the same way as a human. And so one of the things that we've recently done is that we explored what exactly these penultimate neurons in the neural network is doing prior to classification. Is it using histological correlates that we are familiar with, or is it using other features that are completely irrelevant to how a human perceives these different patterns? So here is a metastatic renal cell metastatic colorectal cancer to the brain, and then we wanted to see if these neurons here are perceiving that there is a fibrotic component to this tumor, there is a papillary component or component to this tumor, and there is a kind of luminal structure that's also associated with these gland-like papillary structures. So when we extract these features in this final layer, do they represent stuff that is comprehensible by humans? And you can see here this feature 215, in fact, labels the fibrotic component of this image. Other features label the epithelial component, so this papillary forming structure, which is the tumor. And then other features represent the luminal correlates. So what we think is going on is that, in fact, the computer, when you give it so much information, it's forced to learn very similar patterns to what humans know, and thus allowing you to do these unsupervised analyses and get results similar to what domain experts have really shown is the appropriate way to classify tissue based on their structures. So now we can explore if that's the same case here. Did it in fact pick up the same patterns that are define the names papillary and clear cell carcinoma? So we can take some features that were enriched in the clear cell group. So perhaps some of these features here that you see are high and relatively low in this subgroup, and see if they in fact correlate with the clear cell morphology. So feature 219 was one of these features, and when we find cases and images that had a high activation of, for neuron 219 or the deep learning feature representation 219, we see that the image is largely made up of these clear cells with clear cytoplasm, whereas when you find images in the clear cell carcinoma cohort that had a low activation of 219, you see tumor cells, but these tumor cells have a rich eosinophilic cytoplasm, which is quite atypical for these tumors. So more on that later. So we can do these feature activation maps to kind of confirm that what we're seeing is in fact the case, and you can see that the computer is most activated by the tumor and not by this intervening blood vessel, as you can see here. It completely ignores that. So again, confirmation that this feature likely correlates with the clear cell patterns that we see under the microscope. We can do the exact same in the papillary group, and this is really exciting because technically this is a clear cell feature, so it shouldn't have a correlate in the papillary group. But as you can see, we have a papillary morphology. So this is in fact a papillary carcinoma, but you can also see that the cytoplasm is relatively less eosinophilic than we would expect. And this is actually a newly defined entity. It's a clear cell papillary renal cell carcinoma, a newly defined entity with a good outcome. And you can see that when we go low on this feature, again, you get your typical papillary structures that are much more eosinophilic. So these features are completely portable. You can use them in different tissue types, and again, provide not only transparency, but also histotype type of quantitative tools for digital and cellular pathology. Conversely, we can go into the papillary group and see what features are enriched in the papillary group. And here's one of those features 309. You can see that, again, we go into the clear cell carcinoma group, a group that's not typically characterized as having a papillary feature, but even in the kind of expert annotated images of the TCGA cohort, we get these relatively eosinophilic rich tumors that have some sort of organoid or papillary structures. When you go low on this feature 309, again, you regenerate the clear cell morphology that you would expect in this cohort. And again, we get very congruent results with the TCGA clear cell papillary group, as we see these kind of papillary structures, and then a relatively depletion of those papillary structures in this cohort when you recall images with a low feature 309. So provides a lot of transparency, provides a lot of flexibility into how these decisions are being made. So as we said, that not only is there morphologic heterogeneity, but you can actually use the nuclear features to prognosticate these tumors. So we wanted to see if in fact the neural network could also take one type of tumor, such as clear cell carcinoma, and subclassify it further based on something that would perhaps correlate with these ISUP grading schemes that were recently introduced in 2012. So to test this hypothesis, we took the clear cell carcinoma cases from the TCGA cohort and grouped them based on the patterns of their deep learning feature vectors. And you see this here. We had three major subgroups that arose. And when you look at the survival correlates of these specific groups, you see that they have, in fact, significantly divergent survival outcomes. One group peculiarly has exclusively 100% survival, whereas you have kind of a moderate and much more aggressive phenotype in these different subgroups. So we wanted to explore what perhaps was driving these clinicopathologically distinct entities. So to do this, we again took specific features that were enriched in the short and long prognostically significant group and tried to see if they correlated with something that mirrored the ISUP grading, perhaps, since that also had prognostic significance. So here are some four features that we investigated and characterized further. Two that were enriched in the long-term surviving group, 263 and 254, and two that were enriched in the more aggressive red cluster, 442 and 449. So you can see here are the histogram distributions of 499 and 442, and you can see, as expected, they are increased in the short and depleted in the long group. So we wanted to see if, given the heterogeneity found in the kind of intermediate prognostic survival group, if we could group them based on high and low level expressions. And what you can see here, the 79 highest and 79 lowest patients from this entire cohort, both for 499 and 442, maintain that aggressive phenotype. So again, suggesting that these are now digital histotypic markers that we can histotype patients and predict outcomes, both as a signature but also as independent individual biomarkers. Here are two features that were enriched in the short-term survivors, and again, you see two things. You see again that they're enriched in the long-term survivors and depleted in the short-term survivors, but again, there is a relative heterogeneity within this moderate surviving group. So we wanted to see if these were maintained in larger cohorts outside the proposed classes. So again, we took the top 20 and bottom 20 percentile of the entire cohort and used these values to group these patients. And again, that you see that these survival differences are maintained even at the individual marker level. So what possibly could these represent? So we wanted to explore using feature activation mapping to see if we could find the exact correlates of these unique features. So here is feature 499. Again, we go into the entire database of the clear cell carcinoma group and find images that have a high feature 499 activation and a low feature 499 activation. And what you can see is that the high group has very large nuclei and even some prominent nucleoli, whereas the images that had a low 499 activation had relatively small nuclei. And you can see again that the activation of 499 is within the tumor cells themselves and not the kind of fibrovascular tissue that is present in between the clusters of tumor cells. And so we believe that feature 499 represents ISUP correlate of grades 2 and 3. Feature 442, you can see that when this feature is highly activated, the images show this sarcomatoid morphology that is again depleted. When 442 is not activated, you have more of this ISUP grade 1 type morphology, suggesting that we now have a deep learning correlate of this ISUP grading feature. We can also do the same thing for the deep learning features that showed a good prognostic outcome. So here is 263. You can see that the features, the images that have a high feature 263 look very different than the ones that have a low feature 263. And when you look at the feature activation map, the computer is really focusing on the cystic component of the images, which again is thought to be a good prognostic marker in these tumors. Peculiarly, we had another feature that correlated with good outcomes, which was feature 254. And again, if you look at these images here, you have a relatively eosinophilic rich tumor in the cytoplasm of these tumors, which was reminiscent of a specific benign tumor type called an oncocytoma. And thus, we were interested to see if these were perhaps misclassifications of benign tumors within the clear cell carcinoma TCGA cohort. So to do this, we needed an external approach to understand if these tumors that had high oncocytic correlate were in fact true renal cell carcinomas of the clear cell type, or if perhaps they represented misclassifications of other subtypes with a more favorable outcome. So to do this, we partnered with one of my colleagues, Dr. Trevor Pugh, and his talented research associate, Alberto Leon, and using the transcriptional data in the TCGA cohort, we developed a T-SNE-based approach to classifications of these tumors, with green representing the clear cell carcinomas, blue representing the papillary clear cell carcinomas, and yellow representing the chromophobes. And you can see that these 9 tumors that had a superior prognosis, only 2 of them actually cluster with the clear cell data set. 2, as you can see here, are found very far away from any of these 3 sets. These are presumably oncocytomas. And then the remaining 5 cases actually correlated with the chromophobe transcriptional signature. So what we believe is that these are in fact misclassifications found in the TCGA cohort that account for the superior prognosis survival found in that green cluster. We also showed local pathologists to confirm this, and again, blinded to the RNA transcriptional classifications. They also found that there was a large number of chromophobes that were found in this cohort, and only 2 had features reminiscent of true clear cell carcinomas. So I think that's really highlights how unsupervised approaches are very, very effective for anomaly detection in large databases without having to train a computer on a specific predefined task. So in summary, hopefully you've seen how neural networks and deep learning can provide automation and quantification to cellular pathology. And again, relatively straightforward workflow where you train a neural network and then you can use either the classifications or the feature vectors that often correlate with human constructs to do a wide range of tasks, including annotation, classification, visualization, prognostication, and subgrouping of tumors. I want to end by thanking the members of my lab that have done the hard work for the data I presented today, especially Kevin and Adil, who drove the unsupervised project that we presented today, and all members for providing additional insight and hard work for some of the other projects that you'll see in future lectures. Of course, our collaborators that provided expertise outside the domain of my clinical specialty. So I want to thank Trevor, Alberto, Sangeeta, and Andrew Evans for their important contributions, and of course our funding support, which this work would not be possible without. Very early on, we've gotten funding from the Brain Tumor Foundation of Canada and the Adam Cools research grant to kickstart this work. And then again, more substantial funding from the Princess Margaret Foundation, Conquer Cancer Foundation, and the Brain Tumor Charity in the UK to really continue and expand this work to what you saw today. So thank you very much for your attention, and I look forward to discussing more of our progress in future episodes.